All right. So this is the last portion. Like I said, we're going to start today. We'll finish it up probably on Wednesday. And then hopefully we can start what will be the last chapter. Now we're going to, and I just pick a few, okay? Your book will probably go over even more. I haven't even checked the most recent edition. I picked some of the ones that, I mean, obviously have biomedical implications. I picked another one because I got to use a cool figure, but it's also really important too. So now we're going to see like what happens when you take these amino acids and they're used to make certain molecules.
All right.
Yeah, that's lots of bells.
So for the exam, are we really more focusing on just what you cover?
What I cover or what was in the suggested problems.
Okay.
Because sometimes I deliberately pick the suggested problems that have other medical implications. There's just no way for me to cover everything.
Okay.
And it's 21, 22.
Yes. The last 3 chapters. I think the third one is 23. Okay. So amino acids themselves are used for precursors for lots and lots of different things. And we actually kind of continue. This is one of the reasons why I like to look at chapter 23 because we pick up on that more with the neurotransmitters and stuff. I don't usually focus as much on it now because hormones and neurotransmitters and things like that in the next chapter. So we're going to actually start with glycine. And this one is just, I don't know, poor glycine. You could probably turn this, if I took enough time, think of a way to turn this into a devotion. Because glycine seems so nothing, right? Of the amino acids, it is the most simple. It's the most humble amino acid. I'm going to give it a characteristic, right? Because what's this R group? Nothing. It's a hydrogen. Like its R group doesn't even show up in X-ray crystallography. That tells you how insignificant it is. Yet, for higher eukaryotes, it's what's used to make a porphyrin. Which, what is porphyrin? Do you remember what porphyrins were used for? I mean, it's huge. And it's major important. Without it, you die. What's the porphyrin ring? If we look here, porphyrin rings actually have iron in it. So where did we see iron last semester? Hemoglobin. That's the heme is a porphyrin. Okay, so if you think about it, God has made something that seems so insignificant in the amino acid world, at least, to make something that literally life and death hinges on. Before this, I think the only other thing that we really ever talked about glycine being good for was that being in tight places in protein structure, right? Because it can't be nucleophile, it can't even be an electrophile, it can't even really do beta Volz contacts very well because it's just a hydrogen, but it's actually used to make porphyrins. All right, and so for us, for animals, glycine reacts with succinate, which where have we seen succinate? Citric acid cycle, to form delta aminolevulinate, which is a mouthful, okay? Now in plants and bacteria, they actually take it from glutamate, so lysine, which I'm sure the evolutionists have a heyday explaining that one to you. But if we were supposed to be more evolutionarily advanced, why are we using something that's more sampled than it? But they talk about divergence. All right, and then the iron actually isn't inserted until towards the very, very end. Okay, that's one of the final steps. Okay, and notice protoporphyrin just means it's like a prototype, just like it's another precursor form. That's why I say I'm sure that you can come up with really nice devotion based off of that scientific geek chic devotion based off of this. All right, I don't want you to sit here and just memorize all of these. You can probably figure out the names. Like these aren't way out there. Like delta aminolevulinate synthase, it's a 2-step process, but it literally goes, and all it does is it takes succinyl-CoA from the citric acid cycle with glycine to make delta aminolevulinate. Don't memorize that name. Okay, I would either give you the name of starting materials and the end product and ask you to know the enzyme, or I would give you the enzyme delta aminolevulinate synthase and ask you what it makes. I would assume you'd be able to say delta aminolevulinate. I think that's the game. You can see plants actually, they utilize a different cofactor, a different helper. They use tRNA, but the tRNA is very similar in this instance for what ATP would be. It's just that tRNA is a great leaving group. But so if we're going to do like a compare and contrast, one way to do that is the fact that it uses glutamate, but they also use tRNA instead of, well, we don't even have anything remotely like that on our end. So, okay. Then we have all of these. Do not memorize these. The important thing was glycine is used to make delta aminolevulinate. Then we have all of these proto, all that kind of stuff. And they have, various names. I guess I could point out and say, what class of enzymes? Well, it's decarboxylase, that kind of thing. But that one's actually an oxidase. Number 4 is decarboxylase. But you can see. But once again, I don't expect you to know all of those. But let's play it down to what is important. First off, the iron's not incorporated until the very end. And that's one thing just to point out. Next thing to point out is porphyrin biosynthesis. What it's regulated by is by the concentration of heme. Why does that make sense? If you're going to regulate this, why would the concentration of heme make sense?
Depending on how much you need.
Right. If you've got a lot of heme around for your hemoglobin and for the cytochromes and things like that, you don't need to make any more. So it's going to be a feedback inhibition. There are scads in SCAD. As you can see, if we have all these enzymes and the probability of having mutations in them, just like what we saw with all of the possible genetic mutations of hemoglobin. Remember the blue people? Technically, it's called methemoglobinemia. There are lots of there's the most common, I think of now, sickle cell anemia, and all those. That was with hemoglobin. So you can imagine each step of these, you could possibly have genetic mutations. Right? And these are called the porphyria diseases, since it's based off the porphyrin group. So a little historical side note. I don't usually get to bring in too much history with it. But King George III, he was the king during the American Revolution, and many believe that he was crazy. They thought he was mad. He suffered from porphyria. And so these porphyria diseases, someone calls it, for him, it was abdominal pain, neurological disorders, you can imagine. There's another type of porphyria that results in one of those very specific intermediates that I've already covered up. And once again, I wouldn't ask you to memorize that. But it results in accumulation of uroporphyrinogen 1. It causes your urine to come out red. Your teeth fluoresce. What does that mean?
Shine.
They shine. They can kind of, can you imagine putting that in a black light? They're going to glow. All right? And you become highly sensitive to sunlight. And the reason that they're highly sensitive to sunlight is the fact that they have such severe anemia due to heme production. And so, what do you suppose that could result in? What would you kind of look like? Death? Perhaps. If you think about the phenotype, what would be a good phenotype for this? Typically, what's their skin color going to be?
They're not going to get much sunlight.
Right, so they're going to be really, really pale. They're going to have scary teeth. Okay, and so technically, historically speaking, a lot of people believe that those who are ostracized from being vampires suffer from some types of porphyria. There have been documented cases of people being so abnormally low to heme that they do crave blood. And so that is one of the ways to get it, right? You supplement that's natural supply of blood. So you could.
And they would burn from sunlight too.
They burn from sunlight. They have scary teeth because their teeth glows, and they crave blood. And so this is the talk that she remembered. She was talking about, they were talking about porphyria or whatever. On that note, we're going to end on that little Twilight kind of thing. I don't know. I've never seen Twilight. For me, it'd be Buffy the Vampire Slayer. Okay, then we're going to have to end on that, and I'll go ahead and end class.